Hello viewer, a warm welcome to Elimu TV where you watch and learn. Welcome to the 7th lesson of biology form 4, and today we are going to learn about sex determination in humans. We're still under the same topic, genetics 2. I am your teacher, Madam Christine Okot. So this is what we're going to learn in this lesson today. First, we're going to review the previous assignment. We're going to look at the sex chromosomes. We're going to look at sex determination mainly in human beings. We're going to look at the sex-linked genes, and lastly, an assignment. So let us start by reviewing the previous assignment. Question number 1 was, what type of dominance is portrayed in the inheritance of the blood group A, B? So A, B, both of them are dominant. They will both be expressed in the same way. That is like a 50-50 deal. So the type of inheritance here is co-dominance. Then in blood group A, the genes are either AA or AO. So when there is A and O, A will be expressed more than the O. So in this particular A, we say that the type of inheritance here is complete dominance, because A has more dominance over the other allele which may be expressed. Then question number 2, list all the possible genotypes for the blood group A. So the genotype is the genetic makeup. So the genetic makeup of blood group A can either be AO, A is the dominant, so it will be more expressed, or it can be AA, having both alleles A and A. Then in blood group B, it can either be BO, B is more dominant than O, so the blood group will be B, or it can be either a B and B. So those are the genotypes. To the business of the day, so sex determination in human beings. So humans, we have 23 pairs of chromosomes, or we can say we have 46 chromosomes. So out of the 23 pairs, there are only 22 pairs which determine the characteristics of the body or the traits which can be passed from a parent to the offspring. Now, these chromosomes, the 22 chromosomes, are what we call the autosomes, while the 23rd pair, they are called the sex chromosomes, because they do not determine the characteristic or the trait you're going to inherit. Their main function is to determine the sex or the gender that an offspring is going to have. So that is the example of the chromosomes, the 23 pairs which are found on the human. So we have chromosome number 1 to 22. They're called the autosomes. Those ones are responsible for determining the characteristic which a parent is going to give to the offspring. That might be in terms of the size, in terms of the color of the skin, other things which you can inherit from the parent. Whereas now when we come to the 23rd pair, we have the X and the Y chromosomes. This one, they determine the sex or the gender of the offspring. So there are two types of sex chromosomes in the human chromosome. So the 23rd pair, we either have the X or we have the Y. So the X chromosome is normally longer than the Y chromosome. Looking at the illustration, it's trying to explain or demonstrate the X chromosome being longer than the Y chromosome. So those are the chromosomes which are found, which are responsible for the transmission or determining the gender of the offspring. So looking at the chromosome, the genotype and the phenotype. So the genotype, we've said, is the genetic makeup, and the phenotype is the physical appearance. So if you have the two similar chromosomes, that is the X and the X, the genes, it means that you are a female. If you have the X and the Y, it means you are a male. So these are the only two genotypes which are found in human being. XX denoting that you are a female, and XY denoting that you are a male. You cannot be in between. So how does this determination comes about. So the one who determines the gender is mainly the father, because he has different genes. The mother has the X and X chromosome. So if the father will release a Y chromosome, it means the offspring which will be born will be a boy or a male. While if the father releases an X chromosome from the sperm, it means the child who will be born will be a girl or a female. So the person who determines who the sex of the child is mainly the father, because the mother has constant, the X and the X, and the father is the one who will determine by either giving an X or a Y. So we can have an example to see how this comes about. So here is our example. So we have a father having the X and the Y chromosome. The mother, the chromosomes are the same, the X and an X. So if we do the crossover, you can also use the Punnett square. So in this case, we're going to use a crossover. So the first gene, we have the X with the X chromosome from the mother. So that one will give us a daughter. The second chromosome from the father, which is a Y, which comes, it mingles with the Y, the X chromosomes, it gives a daughter. Then if we have the Y chromosome and the X, the Y chromosome and the X, it gives us a son. So if you are, by having children, the chances that you'll have a daughter or a son is 50-50. So you can either have a daughter or a son depending on which chromosome the father is going to release. So in our illustration here, the chances are always 50% of either having a daughter or having a son. So when you talk of the gametes of the female, they produce both an X and an X. So these are chromosomes of the female. They are referred to as homogametic. Homo means one, so they are the same. Whereas in a male, they produce X and Y chromosomes, which are referred to as heterogamic. Hetero meaning different. So let us linked genes. So we start by first defining what is a link. A link means like to join together or to be bonded together. So the genes are located at the same chromosome. If a gene is located in the same chromosome, we say that gene to be linked to the chromosome. The location of the gene on the same chromosome is referred to as a linkage. In our illustration here, we have the gene and we have the linkage. So if a gene is located on the same chromosome, we say that the gene is linked to the chromosome. So we have sex-linked genes. So these are genes which are found on the sex chromosome. That is either the X or the Y chromosomes. So these genes are transmitted together with those that determine the sex or the gender. For example, if we look at our illustration here, we have A and B. We have the genes. They are located at the same chromosome. So A and B are linked together because they are on the same pair of the chromosomes, or the two chromatids which make up the chromosome. So they are found in the first pair and the second pair. Whereas we cannot say that A and C, they are not linked together, because A is found on a different chromosome and C is found on a different chromosome. So the only genes which are linked here is A and B, and they are linked to the same chromosome. The Y chromosome is normally shorter than the X chromosome, as we look at the illustration there. So the X chromosome, it has genes that have no correspondence allele to the Y chromosome. So the Y chromosome, if you look at the X chromosome, is taller or longer than the Y chromosome. So if you look at the part of the Y chromosome, there are genes which are replicated, or they are the same up to where the Y chromosome reaches. But from part A, B, and C, they are just only found on the X chromosome. They are missing on the Y chromosome. So the Y chromosome is missing that section of A, B, and C of the X chromosome. So the lower section of the both chromosomes, they contain the same genes or the same traits. Now the difference comes between the X has more linked genes, that is A, B, and C, which are not replicated the same in the Y chromosome. So the genes located on the non-homologous part determine different characteristics which are known as the sex-linked characteristics. That's why we say most of the sex-linked characteristics or the genes, they are located at the X chromosome. So the upper part, the one which is missing, that is where it carries the sex-linked genes. So we have example of the sex-linked traits that we're going to look about. So first we have the color blindness, we have hemophilia, and we have either hairy ears and nose. These are the genes which are found at the sex chromosome. So an illustration here, we have a hemophilia, a condition whereby there is a low level of blood clotting. So someone loses a lot of blood due to the malfunction of the platelets to produce what is used for the clotting of the blood. So let us look at the first sex-linked gene, which we call the hemophilia. So what exactly is this hemophilia? This is a disease which is caused by failure of the blood to clot or take long time to clot. So for example, you might be having a cut and you discover that maybe you bleed for a long time, or it takes more time for the blood to clot so that the bleeding can stop. So this condition is what we refer to it as hemophilia. So in hemophilia, this is a sex-linked gene, mostly on the X chromosome. One allele will determine the clotting of the normal clotting factor, that is the dominant allele, whereas the other allele influences the information of defective clotting factor, which is the recessive allele. The dominant allele is represented by a capital H to show that it is dominant or superior, or it is suppressed more, whereas the recessive allele is represented by the letter H. So these are some of the conditions, or this is what happens when you do not have the platelets that help in the clotting of the blood. Sometimes you can be bleeding externally and it takes longer for the blood to clot or to stop bleeding. Sometimes the bleeding is internal. So let us have a video that is going to help us explain more about this genetic linked disease which we call the hemophilia.
The results from your blood test that you did last week came in yesterday, and I'm afraid to tell you that you have a rare form of haemophilia. It is a genetically inherited disease which affects the ability for blood to clot. Haemophilia does not affect the rate of bleeding, but rather prolongs the effects of bleeding.
How does the disease work?
To explain that, we have to delve deep into the world of DNA and genetics. DNA stores the genetic information responsible for creating new proteins and new cells. It stores this information in the classic double helix shape, consisting of the backbones, the bases, and the base pairs. The whole DNA helix shape is then stored in the nucleus of the cell. Genetic information is data that describes a person's traits. These traits include a person's gender, race, weight, height, and the person's genetic inheritance. This information is stored in the form of different chemical combinations, which is much like how a computer binary works. DNA is key for an organism to develop, survive, and reproduce.
Oh, so it is the structure of the DNA that caused my hemophilia.
No, it isn't the structure that causes haemophilia, but instead a mutation in one of your genes.
Oh, okay.
Look, I'll show you.
A point mutation is when the DNA breaks and a base pair is repaired, however the wrong chemical is chosen. This phenomena occurs during reproduction, when the daughter cell notices an issue and corrects it using its repair enzyme. The problem really takes place when the original leading strand's chemical is replaced rather than the newly created copy. For example, an adenine base joining with a cytosine base forming a mutated DNA component, or in this example shown, yellow to green. This therefore creates an issue when the cell is replicated, and the newly created cells also have the defection. An insertion mutation affects the length of the DNA on one side when compared to the other. This is caused by a break in the DNA chain, which could be contributed by outside factors such as radiation and chemicals acting upon the delicate DNA. The repair enzymes repair the broken DNA by adding extra nucleotides to the chain, which ultimately lengthens the backbone on one side, leading to complications during reproduction. A deletion mutation is when the segment of a DNA strand is lost or broken, and the repair enzyme simply joins the two connected ends without replacing the lost chemical instruction. A loop is then formed in one of the strands, and when the first cell is replicated, one of the produced cells gets less information than the other because it wasn't included in the reproduction stage.
Ah, that makes sense, it is a genetic disease. But what is hemophilia exactly, and how does it relate to mutations?
Okay, let me explain. Hemophilia is caused by a mutation in a single gene that is altered or missing. There are two main types of hemophilia, A and B. Hemophilia A is the most common form of the disease and is found in 1 in 4,500 males. In order for the disease to occur, a mutation in the F8 gene needs to happen. The F8 gene is important to produce the coagulation factor 8 protein, which works with platelets in the blood to help control clotting. Hemophilia B is much the same type of mutation, however it is less common, more serious, and is a defection of the F9 gene. The F9 gene is responsible for much the same protein production, however the protein is called coagulation factor 9. The second form of hemophilia is much rarer, with only 1 in 20,000 being affected.
So do I have hemophilia A or B?
You have been diagnosed with hemophilia A.
Okay, how did I get it?
Let me explain. Well, Liam, hemophilia, as discussed earlier, is a disease which is usually passed on genetically. This has led me to infer that your mother must have been a carrier of the disease. Due to your mother's genetic makeup, she wouldn't have shown any symptoms because she has 2 X chromosomes, meaning one would have cancelled the other out. The diagram shows that you had a 50% chance of developing the disease because you're a male. So I presume from my knowledge that your mother was a hemophiliac and you inherited the gene mutation from her.
What options do I have for treatment?
There is one treatment which is currently universally accepted amongst the medical community. That is replacement. This involves the injection of the clotting factor into the bloodstream. Many clinical trials have been undertaken about the future cure to the disease called gene therapy, which would work by producing artificial microchromosomes filled with DNA. The patient's cell, being removed from its body, would then be inserted with the chromosome and the cell is cured of hemophilia, because it has the instructions needed to create the protein. The cell then multiplies and is injected into the patient's bloodstream, where it spreads throughout the body and creates the coagulation factor.
Do you think this cure will be available in my lifetime?
The research is promising, however the cure is still a long way away and is still undergoing clinical trials. There is a hope that it will become available for commercial use sometime in the not so distant future.
There is hope.
So I hope you have enjoyed the video, and apart from enjoying, you have learned 1 or 2 things about hemophilia, about how hemophilia can be treated. So let us look at some of the crosses within how you can inherit this hemophilic disease. So we have a cross here. So how is it first represented? If it's a normal male, it is represented by H. H is the allele for the dominant clotting factor. It means that if you have the capital H, you do not have hemophilia. So for a normal male, it is the X that has the capital H. In a hemophilic male, it is the X. Remember, it is carried on the X chromosome. So the X bears the small h, meaning that you are hemophilic. So in male, it's only 2 traits. It's either you are a normal male or you are hemophilic. In female, it's different because you have 3 parts. It's either, if you are normal, it means that you carry the capital H on both, say, X chromosomes. For a normal female, but he is a carrier, it means that you're not going to see it, it's not going to show on the phenotype, you're not going to recognize it in the terms of physical appearance, but in the genetic makeup, it shows that she is a carrier. So she's going to have both the capital H and the small h, denoting that she is a carrier. Then for a hemophilic female, it means that you have both the small h, showing that you have both the hemophilic genes. So an example of a cross between a normal female and a carrier female. So the phenotype, this is the physical appearance, this one is a normal female and the other one is a carrier woman. Then the genotype, the genetic makeup, as we have said, if you have the capital H, it means that you are normal. If you have the small h, you have the hemophilic disease. So this one, the normal male does not have, it means he's carrying the capital H. The carrier is carrying a capital H and a small h. Now let us do the crossover. The first crossover is going to be between the first X chromosome of the male and the first X chromosome of the female, producing both of them. They will carry on the capital H, normal female, XX for the female, and the capital H denoting that the person does not have hemophilia. The second cross is between the second chromosome of the male, the Y, and the first chromosome of the female. So it will produce an X for the female and a Y. Then he's carrying the capital H, that meaning it is a normal male. Then looking at the third cross, we have a normal, the male, with the female, the second X chromosome, which has the capital H. So this one is going to be a carrier. Then finally, we have the last cross of Y with the second chromosome of X, which has the hemophilic. So it's going to produce a hemophilic male. Remember, in a male, there are no carriers. You are either having hemophilia or you are normal. So you're going to have the phenotype, a normal female, a normal male, a carrier female. Let us look at another example of a cross. So another cross is between a normal male and a carrier female. For a normal male, the H is going to be capital, denoting that he does not have the hemophilic gene. For a carrier female, is going to have both, but one is going to be, the H is going to be capital and the X the small, because she is a carrier. Now, when you do the cross, the first one, the X and the X with both H, it means that the female is normal. You do the second cross, the X, still looking at the X and with capital and the X with a small h, it means this one is going to be a carrier because she's having both the normal and the one for hemophilic. So in the phenotype, it's going to show that she's normal, but on the genes, the genetic makeup, it shows that she is a carrier. Going to the Y chromosome with the X chromosome, the first one is going to give us X with the capital H and a Y. That means that the person is a female who is normal, does not have hemophilia. Then the second cross of X with a small h that has the hemophilic gene, and a Y, this one is going to be a male who is hemophilic. So, and another example as a cross between a carrier woman and a hemophilic man. So a carrier woman will have both capital H and a small h, and the hemophilic man will have the small h, denoting that he has the hemophilic gene. So when you do the crossover, first you'll get a carrier girl who has the small h and the capital H. Then you'll have a normal boy that does not have any of the hemophilic gene. Then the third cross, we're using the Punnett square, we're going to have a his. So when you're talking of color blindness, especially the red-green color blindness, this is a form of color blindness in which a person has trouble seeing any pigment or any shade of the color red and green. So in our illustration here, we have the colors that somebody can see when he has the normal vision and the color somebody can see when he has the color blindness. For example, if you're looking at red, somebody who has the red-green color vision, he will not be able to visualize that is red. Rather, he will see something else like a shade of brown or something else. If you look at green, you will also be not able to see the green color, but you will see something else. So this person is not in a position totally to visualize anything that has red, anything that has green, or anything that has a shade of both red or a shade of green. So this is how color blindness is represented when we're doing the crossing over or when we're using the Punnett square. In the female, we have the 3 genotypes. So if the person is normal, he does not have color blindness, we use the capital C on both the X chromosome. If the person is a carrier, it means that he will bear the capital C and a small c, capital meaning that he does not have, and the C meaning that he has the color blindness. So that one will be a carrier. Then still on the female, the last genotype is both of them bearing the small c, meaning that they have both the color blindness gene. Going to the female, so in female there are only 3 possible genotypes, while what you can see, the phenotype is only 2. If somebody is a carrier, you will not be able to see in the phenotype. In the male, we only have 2 possible phenotypes. If you do not have color blindness, the X will bear the capital C sign, while if you have the color blindness, you'll have the small c at your X chromosome. So there are only 2 possible. You are either colorblind male or you are a normal male. So let us look at some of the cross of color blindness. So the first one, another cross is between a colorblind woman and a colorblind man. So all of the chromosomes will have a capital C on their chromosomes. So if you do the crossover between the colorblind woman and a colorblind man, all of the offsprings are going to be colorblind. For example, you have 2 colorblind girls and 2 colorblind boys. So all of them are going to be colorblind because the woman is not a carrier, but she is colorblind. So all of the offsprings are going to be colorblind. So this is a cross between a carrier female and a colorblind man. So if you do the crossover, the phenotype is the one who is a carrier female, then another one is a colorblind man. The genotype, one of the chromosomes is going to have a capital C and another one a small c, denoting that it's a carrier. The male one is going to have a small c at the X chromosome. So when you do the crossover, you'll have a carrier daughter, a normal color vision son, you'll have a colorblind daughter, and finally you'll have a colorblind son. So another example of a cross, but this time using the Punnett square, of a carrier woman and a colorblind man. So if you do the crossover, first of all, the genetic makeup is going to be, for a carrier, you'll have the small c, and for a colorblind man, the small c on the X chromosome. So when you do the crossover, you'll have a colorblind girl, a carrier girl, a colorblind boy, and a normal boy. So the question here is it possible if a carrier woman and a colorblind man marry, is it possible for them to have a normal boy? The answer is yes, because we have seen the crossover here using the Punnett square. So for your assignment today, question number 1, state the possible genotypes of a normal person in relation to hemophilia. Question 2, on which chromosome is the gene that causes hemophilia linked to? Question 3, what is color blindness? So you're supposed to define what color blindness is. Question 4, what will be the genotype and the phenotype of offsprings between a carrier woman and a hemophilia hemophilia and a normal male. So you can either use the cross test, or you can use the Punnett square. So that has been our lesson for today, and you can pose your questions or any feedback. Our Facebook page is Elimu TV. Our Twitter handle is at Elimu_KE. Our SMS lines is 22518.